Okay, so the topic for today is phylogeny. And for this story, I'd like to start off with our human tendency to want to organize and describe nature. Now, this is something we've been up to for pretty much forever, but I gotta start somewhere, and one point where I chose to start is with our Swedish Carl von Linné, who systemized nature and gave us our start of our modern taxonomy, where we're organizing things with species, genus, families, and all the way up to kingdoms. Now, having sort of put things in boxes, it's quite natural that we want to illustrate this, and a tree-like description of how this sort of thing fit together was quite soon to follow. But to sort of get from there to our phylogenetic trees of today, where we're indicating history and genealogy, we needed the addition of Charles Darwin and the theory of evolution. That sort of brings the foundations together. So with phylogeny, we're looking at evolutionary relationships. So how did we develop from a pre-existing form to the current form? And phylogenetics is the study thereof. Now, one term that's gonna show up quite a lot is phylogeny, and that specifically refers to the evolutionary relationship between organisms. So this phylogenetic tree here is a branching diagram that's describing the phylogeny between these different organisms that are displayed in the tree. Now, this is something that can be quite useful. So one example would be with how do we sort an animal? Where does it belong in our taxonomic framework? So the giant panda was for a long time a bit of a riddle, because it looked like a bear, but it also had features that are not that bear-like. They're more raccoon-like. It doesn't hibernate, for example. And that riddle got its solution in 1985 thanks to molecular phylogenetics, where we're looking at either genes or protein sequences to see how are things related, rather than looking at morphological or observable characteristics. And with this, they could build a phylogenetic tree where it's clear that the giant panda is indeed within the bear branch in the taxonomy, but it was one that diverged earlier, and thereof the confusion. So this is one typical example of where it can be handy to use our molecular approaches. And what we're actually doing, like, we could look at the full genomes of the organisms, but this is possible. It's commonly not very practical at all, because you're just dealing with such a large data set. You have lots and lots of positions to compare, and that's going to be time consuming, and you may also have accuracy issues if what you're comparing is too far apart or too much noise from non-coding sequences, etc. So what we're actually doing in practice is to build the phylogenies based on a set of genes rather than the full genome. Now, that means that we have to select these genes, and some of the genes will evolve faster and other will evolve slower. So to sort of deal with this, it's quite common that you're looking at a set of genes, and you quite often want to use a variety of gene families to even things out. And the phylogeny that you're building, it's actually reflecting the evolutionary history of those genes that you have built it on, rather than the actual evolutionary history of the species themselves. That is something that we infer from their genes. And one type of gene that's very common to be used for these purposes is ribosomal RNA genes, because they tend to have an appropriately slow evolution to be informative for species. And it is indeed the 16S rRNA that's the predominant way of constructing phylogenies of the prokaryotic organisms. So that's one direction. Now, let's have a look at the actual workflow. How do we go from wanting to construct a phylogeny to actually having the final phylogenetic tree? Now, there are a few sections here that we're gonna go into today. First off, we have sort of the pre-work that we've touched on. So it's your selection of molecular markers. Using them, you construct a multiple sequence alignment. Then you also need to select a model of evolution to sort of handle some of the quirks that are in action at the molecular level. Having done all of this, you can go the way to the next step, which is using a tree construction algorithm. And that's what actually gives us our phylogenetic tree. Once having the tree, we need to assess, is it a good tree or is it not? Are there better trees? How confident are we in what we have constructed? But what better place to start than in the beginning? So to sort of understand some of the choices that we need to do here in the preparatory steps, we need to look at evolution at the molecular level. And one of the predominant mechanisms through which this happens is mutations. So let's bring up an example. Here we have a few things that are a few sequences that could be DNA codes, and then we see that we've changed from a G to a T and from an A to a C. And these are substitutions, so when we've changed from one base to another. And these DNA sequences, they encode proteins, the things that do biology on the next level. So every 3 of them encode a amino acid. And that's what I have a nice table of here that we're gonna put to use to see what effect have these substitutions had on what's encoded by these sequences. Now let's look at the first one. So we have gone from C, C, and G. So C, C, and G, that's a proline. And C, C, and T, that's also a proline. So we haven't had any change at the amino acid level. So this is a synonymous substitution, meaning that it's a change at the DNA level, but not at the protein level. Now let's have a look at the second one. So our current sequence is T, T, and C. So that's a phenylalanine. But before, we had T, T, and A. T, T, and A. So that's a leucine. We've had a change at the amino acid sequence, at the protein level. So this is a non-synonymous substitution. Other things that can happen to our code is insertions when a new base is added in, or deletion when we're losing a nucleotide, and these can affect the reading frames and have loads of impacts as well, or maybe not, who knows, it depends. But what we actually will observe, that's something that's determined by selection. So which changes were successful, propagated to the next generation so that we can observe them so that they can continue to evolve. Now, there are a few different concepts we're going to bring up here. So first off, we've got the neutral evolution, and these are changes where there's not really any selective pressure. The change is neither beneficial nor detrimental, so it's sort of not doing much in the end. And this gives us the rate of amino acid change that's expected from chance, our null frequency, so what we expect from just pure chance with regards to change. But what happens when we're not just randomly changing things, when there is an active selective pressure? Well, there can be negative selection, and that's when a change is detrimental. It's bad for the organism who had it, and that means that it's going to propagate less, so fewer offspring carrying this detrimental change. So we're going to be observing bad changes less often than neutral changes. So we're going to have some frequency that is lower than the null frequency. Now, we'll see this as positions that stay the same for longer than expected. So we call these positions conserved because they're conserved over time. Finally, we have positive selection, which is also sometimes called adaptive evolution. And that's when changes are beneficial and they end up being propagated. And that means that it's good for them, and we see it more often than null frequency. At the amino acid level, then we'll see more changes that are non-synonymous, because this is the type of change that will contribute to evolution of new functions, and that tends to indicate that you need to have a change of amino acids, because that's where biology happens. So there are, of course, a few more ways that genes and genomes can change and affect through evolution. So we have gene duplication events, where we had one gene through a number of possible processes. We ended up with 2 copies of the same, and these copies can then independently evolve, and maybe we'll have some losses of function in one but not in the other. They're independent, but they have a shared origin. There can also be gene fusions, when we go from having 2 separate genes to being one and the same gene. And horizontal gene transfer, when we have transfer from one organism to another that's not happening through a parent-offspring route. Knowing all of this, our choice of what to build our multiple sequence alignment on depends on what we want to do. What are we looking at? What are we looking for? So if we're looking at something that evolves quickly, we want to have molecular markers that are also evolving quickly, so that they're sort of in pace with the change that we're trying to capture. If we're looking over change at long distances of times, we want something that evolves slowly so we can resolve it. Now, one of our fundamental choices we have is whether to use a nucleotide sequence or a protein sequence. They both have their beneficial sides and their drawbacks. So it will very much depend on what we're trying to do, which one is appropriate to choose at any one moment. So the nucleotide sequences are more rapidly evolving than protein sequence. So as we saw before, we have synonymous changes within the nucleotide sequences, so there's some redundancy there before we actually see it have an impact on the protein level. But if we're looking at something that's very closely related, so say individuals within a population, or track something that's very rapidly evolving, such as a virus, then it may very well be appropriate to look at the nucleotide sequences just because they are evolving more quickly. But in most cases, it will be appropriate to be looking at the protein sequences for a number of reasons. But we do have options for more slow sequences for the nucleotides as well, but for the most distant relationships, conserved protein sequences are probably the way to go. Insertion and deletions can be quite problematic for the nucleotide sequence alignment. We're dealing with just 4 characters in those sequences, so it's hard to align them correctly because you could end up with things that match quite well by introducing a insertion or a deletion in the wrong position. Whilst having 20 amino acids to deal with for the protein sequences, that really does make it more sensitive in the alignments. So we have a few assumptions that we sort of think need to be true or treated as true for the phylogenetic tree construction to be successful. So one of the core assumptions here is that each position in a sequence is assumed to have evolved individually. So we're just looking at one position and it should be unaffected by its neighbor. Doesn't necessarily hold true, especially at the nucleotide level, but it's still a reasonable assumption for the most part. We also assume that the positions that we have aligned are actually genealogically related, that they do have a common ancestor somewhere. And that also relates to the fact that we need our sequences to be homologous, and that means that they need to share a common origin for our phylogenetic tree constructions to be valid, because we're trying to infer the relationship over time, over evolution, and for that to be meaningful, they need to actually be related to each other. Now, there are a few words in this region that we're going to bring up so that we're aware of them and how they relate to each other. One of them is paralogs, and that is homologs that have arisen from a gene duplication event, whilst orthologs are homologs from a speciation event. So close, but slightly different from each other. Then we have analogs. These are genes or proteins that are not related to each other. There's no common origin, but they do fulfill similar functions. So a little bit of convergent evolution going on. Finally, we assume that the variability among the sequences is sufficiently informative for us to be able to construct unambiguous phylogenetic trees. So they need to be informative. There can't be too much difference that's too noisy for us to use, or too little difference that we can't really tell. They're all pretty much the same. So we sort of need it to be in that informative range. So there are a few more things that can happen if we let time run its course. So we can have multiple substitutions. So over time, it may be that we have an A that goes to a T, that goes to a G, that goes to a C. But if we're looking at it, it may look as if we just have a C. We can't tell what's been going on in between. Similarly, we can have convergence, that we're observing a G, but it has indeed gone from one thing through something else to the same. So this is something that we need to deal with, and this is where I called the evolutionary models in my overview comes in. So our definition here is to compare homoplasy, which is similarity that has been independently acquired, that we need to deal with our evolutionary models, and homology, which is similarity due to shared ancestry, which is what we're trying to use in the phylogenetic tree construction. So here we've got our ancestors that have evolved independently to the same thing. So with homology, these are similar. We would assume that they're homologous, but in fact we're dealing with a homoplasy, since we magically know our ancestors, because this is an example. In reality, we tend to not know the ancestral sequences. That's where our substitution models, our statistical models, come in to correct for this occurrence. There are a few different models. I'm just going to mention a couple of them. So there's the Jukes-Cantor model, and here we assume equal probability for substitution between all of the nucleotides. Here's the math. So it calculates the evolutionary distance between two sequences using the proportion of substitution. So how many differences did I have in how many characters? Well, thing is, all of the substitutions don't happen at the same rate. So in a more advanced model, we allow for different substitution probability between transitions and transversions, and that's words describing what sort of change we have between the nucleotides. So transitions are between nucleotides with the same ring structure, whilst transversions are between nucleotides with different ring structure. That lends itself to this nice piece of math, where we're again calculating evolutionary distance, but we're allowing instead of just looking at the proportion of substitutions, we're looking at how frequently do we observe transitions in our sequence alignment, and how frequently do we observe transversions. Now, sometimes we're dealing with protein sequences, and then it's quite common that we're using substitution matrices. So the same sort of thing that we were using in the multiple sequence alignment part of the course to score how good our alignments were. There are related matrices or versions of the same ones that may be used to correct for homoplasy within protein sequences. With that, we are ready to move on to the next section. Now we're going to save the tree construction algorithms for a bit and head into the lovely realm of dealing with all of the words related to our phylogenetic trees, so that we can describe them and talk about them and everything. So first off, we have our topology, and that is the branching pattern of the tree, and this consists of branches. Here we've got an external branch mark, but if we're just looking at something like this, it would be an internal branch. If we track something through the tree, then we have a lineage. Now, all of the points where branches join are called nodes, and we have a few different types of nodes. At the very end of the branches, we have our taxa. Well, it may be that we're not actually having a true taxa there, but we're having a specific gene. So the correct way to refer to these are operational taxonomic units. So that's the most general and inclusive term to describe what we tend to put on our branches. And these are species or sequences that exist today, so are observed sequences. Within the tree, we have internal nodes, and these represent speciation events in case we're dealing with a species tree, or gene duplication events if we're dealing with a gene tree. Finally, at the very bottom of the tree, we have the root node, and this is the node that represents the common ancestor for all of the members in the tree. Then we have a few more words. So we have paraphyletic groups and clades, which are also called monophyletic groups. Now these are groups that comprise of all linear descendants of their common ancestors, whilst the paraphyletic groups, they're a subset of a monophyletic group. So some, but not all, of the descendants of a common ancestor. Final set of words for this slide. We have different words describing the branching pattern of the tree. So if a node splits into two branches, it's a bifurcating node. And if we have a section of the tree where we only have bifurcating nodes, so all of the branches are in two, it's a dichotomy. And that can be contrasted with a multifurcating node, where we split into more than two branches, and that then gives us a polytomy, where we have more than two branches from a single node. So a polytomy can arise either from us not having sufficient data to resolve the tree into dichotomies, or it can actually arise from a radiation event where we have a common ancestor that have more than two direct descendants. So the general assumption, however, when dealing with phylogenetic tree is that we are supposed to have a dichotomy for the entire tree in the end, so only two daughter branches when splitting. There are more ways to represent trees than just the one. So first off, we can use either angled ways of drawing the trees or a square form, and these are equivalent. Branch length, however, that is something that gives rise to two different types of trees. So we have cladograms, where the branch length is not proportional to the evolutionary distance. And then we have phylograms, where the branch length is indeed proportional to evolutionary distance. And this is the more informative type of the two. They're often called scaled trees, since the branches have been scaled to represent the evolutionary divergence. Rooted and unrooted trees. So I did introduce the root, and that is the common ancestor to all of the taxa that we have in the tree. And when we have a root to our tree, then the evolutionary paths that we can draw through the tree indicates directions, and this means that these are indeed more informative than if we don't have a root. So here we have an example of an unrooted tree, and here we have fewer assumptions. We're not assuming anything about the common ancestor. We're just showing the relative relationship between taxa, but any evolutionary path that we try to draw from it doesn't have any direction. Now, if we do have an unrooted tree, but we do want a rooted tree, there are a few methods of going from unrooted to rooted. One of them is the outgroup rooting approach, and then from external knowledge or being familiar with your sequences, you have chosen to include a taxa that you know is more distant from the other ones that you've included, and that will be your outgroup. So say that we know that A is more distant from B, C, and D than they are from each other. Then we can sort of set our root on the branch between A and our internal node and bend it into a rooted tree where we have A as the outgroup. Another method is the midpoint rooting approach, and here you're choosing the midpoint between the two most divergent taxa as the position of the root. This involves the assumption that the branch length is proportional to evolutionary time, and that's called the molecular clock hypothesis, and that's associated with an uniform evolutionary rate. If we applied this method to our unrooted tree, we would place the root in between, well, like right in the middle of this tree, because all of the branches are the same length, and end up with something more like this. Well, if we're dealing with loads and loads of sequences, lots and lots of data, it's impractical to do things by hand. We need our computers to be able to understand these trees, to represent them, to store them, to draw them from a file. And graphical representations are great for us, but not so much for computers. They tend to deal with text better than figures. So could we just transform these into text, something using characters we have on our keyboards? So maybe we should represent our trees using ASCII art? Well, it's not impossible, and it may indeed show up as a way to represent trees ever so often, but to actually communicate a tree to a computer, it's quite common that the so-called Newick format is used. And this is a format for representing phylogenetic trees that is computer readable. And we can do these by hand, but you gotta keep track of all the parentheses, because that's what we're using to define the groups and separate the groups. So let's see if we can transform these trees into the Newick format. First, we identify the group that's closest to each other, in this case B and C. We group them together, parentheses and commas. Then we move down the tree to see what's the next one that's close by, and that would be then A. So we add another parenthesis around all of it and add that A is closest to the BC group. Moving down the tree, we hit the root and we have the other main branch that we're then going to fill in, and that's, in this case, just D and E, which gives us our final Newick representation of this tree once we add a closing parenthesis around the entire thing. There we have it. You can also add branch length to this representation, and then you're using a colon to add the branch length right after the thing that the length should be applied to. So if we use our second ASCII art tree to apply the different branch lengths, so one pipe equals one length unit, we end up with something like this. So B is one length unit away from the closest node, C is 2 length units away, the BC group is 1 length unit away from the next node, and so on. Now, if we're going to make all of the possible trees, we're gonna be making loads and loads and loads of trees because it rapidly grows. For tree taxa, there's just one possible unrooted tree, only 3 possible rooted trees. Moving up to 4 taxa, for the unrooted we go all the way up to 3, whilst for the rooted ones we go to 16. This will give us a rapid rise in the number of possible trees as the number of taxa increase. How rapid, do you say? That's what we've got these lovely pieces of math to explain. So we've got one formula for the number of unrooted trees which are possible, and we've got one formula for the number of rooted trees. So this takes then the number of taxa and uses factorial. So these formulas, as far as I can tell, falls out from combinatorials and math and the fact that we're assuming that things are bifunctionating. And that means that we're going to have to use factorials. And a factorial is multiplying each of the numbers up to the number that we're having a factorial of. So the example here is the factorial of 3 is 1 times 2 times 3. If we had the factorial of 4, we would have 1 times 2 times 3 times 4, and so on. And that's something very commonly seen in combinatorics. And given these formulas, you can then calculate how many trees are possible given the number of taxa that I'm trying to build a tree for. And this is also why when we have lots of taxa, we're going to need lots of computer power and quite possibly smart approaches to constructing our trees rather than evaluating all of them, because there's gonna be an upper limit to how many trees we can feasibly look at just due to computational time and power. But what to do if we have several trees that are equally good? That's when the concept of consensus trees come in. So say we have these three trees that are presumably equally good. We want to construct consensus trees. There are a few ways of doing it, and one of them is to use strict consensus. In this case, for us to assign something unambiguously, we require it to be present in all of our trees that we're using for the consensus trees, and any location where they conflict, we create a polytomy. The other approach is majority rules. So if more than 50% of the trees that we're using agree on a topology, we're gonna use that. If 50% of them don't agree on it, then we're gonna have a polytomy. With that, we have enough phylogenetic tree vocabulary to move on to the different tree construction algorithms. So there are a few different families of algorithms. So we have the distance-based methods, where we're using a distance measure from the pairwise alignment to construct our trees. And there are the character-based approaches, where we're constructing and considering what the ancestral sequences are likely to have been. We're going to start off with the distance-based methods, and specifically with the clustering-based methods. And these are methods where you're joining the most similar ones step by step and eventually ending up with a tree. But to do so, we need to have some sort of distance measure. We need to know how far away our sequences, and therefore our taxa, are from each other. This is represented through distance matrices, and that's a table with evolutionary distances between all of the pairs. So I've grabbed a short snippet of protein sequence from the spike protein from our SARS-CoV-2, the original SARS virus, MERS, and from a bat coronavirus. And that's going to be what we're using for our example. Now, these are the sequences we need to compare to calculate some sort of distance measure. But what is a distance measure for sequences? There are multiple possible options. Sort of the most naive one, or easy to do, is Hamming distance, and that's just counting the number of mismatched positions. Ramping up the difficulty level, well, it's not all that hard, at least not yet. It's looking at the fraction of mismatches. So how many differences do I have divided by the number of positions I have in total, so the fraction of mismatches. More advanced, or we have substitution matrix-based approaches, and this is typical for protein sequences. So there are matrices that, well, it's sort of the same scoring matrices as for multiple sequence alignments. They're typically calculated based on looking at a cohort of sequences and seeing how commonly you see each type of change, and then that can be converted into a matrix that can be used for scoring distances between sequences. Similarly, there are more analytically based models, so we have the Jukes-Cantor model and the Kimura model. The examples of these in the books show the math for the nucleotide sequences, but there are also protein sequence versions of them. So for my example, we're going to look at the fraction of mismatches. So we're going to pairwise compare all of these sequences to fill in our table. So first off, let's look at SARS-CoV-2 and SARS-CoV-1. And we have 4 mismatches, we have 16 positions, and that gives us our distance. Then we're comparing SARS-CoV-2 to MERS coronavirus. We have a few more mismatches. We have a new value for, or we have their distance. Finally, we compare to the bat coronavirus. Now we've sort of filled in all of the boxes that are related to SARS-CoV-2 and its pairwise distances. Then we move to look at how SARS-CoV compares to the other ones. So SARS-CoV versus MERS, and SARS-CoV versus the bat coronavirus. Finally, we compare MERS to the bat coronavirus and get our final distance to our finished matrix. And with that, we can move on to our first tree construction algorithm. And oh, right, forgot this one. So with the distance matrices, there is a concept called an additive matrix. A matrix is additive if there is a tree that exactly fits the matrix. So the branch length will match to the distances in the distance matrix. This is quite commonly not the case. Sometimes you can be close, but it's rare that you end up with a truly additive matrix. So, I promised you my first method, and that would be the UPGMA method. And this stands for Unweighted Pair Group Method Using Arithmetic Averages. The approach is so that you start off with identifying which pair has the shortest distance between them. So looking at our matrix, that's between SARS-CoV-2 and SARS-CoV. So that's what we're going to select and start off with joining these two into our first little beginning of the phylogenetic tree. Having done so, we need to calculate how long are the branches. And this is then an arithmetic average. So we think that the branches, well, let's make them half of the full distance. So if we go from SARS-CoV, we're traveling half of the distance to get to the node, and then the second half to get to SARS-CoV. With this done, we can update our distance matrix so that we can calculate, or so we can identify what's the next thing that we're going to add to our tree. And that's where we're using the average distances to the joined taxa. So we're going to treat the taxa that we joined as a new single taxa. And then to see what are the distances to our new taxa, we're going to take the averages of the distances to the old. So for the bat coronavirus, we had a distance of 0.5 to SARS-CoV-2 and a distance of 0.625 to SARS-CoV. So the distance from the bat coronavirus to our new taxa A is the average between these two values. And with that, we can update and have our new distance matrix that we can carry on with to construct our tree. So, looking at this new matrix, we can identify that the bat coronavirus is closely related to our merged taxa A. So that's the next thing that we add to our tree. We calculate the branch lengths, and that's half of the full distance to A. So we've got that added in. Let's treat bat coronavirus and A as a single taxa B and calculate how far away is the MERS coronavirus. Then we're looking again at the average distance of MERS to A and of MERS to coronavirus, or to the bat coronavirus. And then we get our new value and can finish up our tree by calculating the final branch length. And there we have how these sequences or sequence snippets, because this I've used such a tiny amount of information that this is probably not very representative of the real world, but who knows? Maybe we landed somewhere close. So the last group that we added in a UPGMA tree, that's treated as an outgroup. So we're ending up with rooted trees by using this approach. However, we're also assuming that there is a constant evolutionary rate, so the molecular clock hypothesis is in action. So all of our taxa are going to end up equidistant from the root. But it may well be that things aren't evolving at the same speed. So this is a bit of a shortcoming with this method. Also, we can see that it's very common that the distances calculated for the sequences is different from the branch length calculated for the tree. And then we can very much see that the matrix wasn't additive and that the branch lengths in the tree are not necessarily one-to-one with the sequence distances. Next up, we have our second clustering approach, and this is the neighbor joining approach. This is the most common distance-based method for phylogenetic tree construction. It's very common and it's very good if you have lots and lots and lots of sequences that you need to merge. It's fast and it has a number of benefits. One of them, compared to the UPGMA method, is that it does indeed allow for varying evolutionary rate. However, it gives unrooted trees, so we need to do a little bit of rooting to get to that extra informative level. But it will produce trees with the shortest evolutionary distance between taxa. It will produce a single tree, not multiple ones. However, this single tree, if we have an additive matrix or a matrix that is close to additive, it is the best tree possible. It does hang on the fact that the matrix need to be close to additive, and it can very much happen that it's not. So it's a bit of a risk with it. The algorithm itself functions by starting off with a star-like topology where all of the sequences are joined to a single node, and then you're one by one take them out, creating internal nodes to give the minimal sum of branch lengths. And if you want to look into the details of the algorithm, I would recommend Wikipedia over the course book. It's a bit more clear, the different steps that they go through, and on these sort of very technical subjects, Wikipedia is quite often a very good source and more up to date than older books. So that's, if you want to dig into this, that's the place to go. Now, since it's maybe that we're just creating a single tree, it's possible that we're missing the best tree, especially if we have a situation where there are multiple equally good options and the main algorithm will just pick one of them. This has been handled in the generalized neighbor joining approach, which allows you to consider multiple initial trees and get more topologies to compare, and therefore alleviating some of the risks involved with getting just a single tree from the method. The second type of distance-based approach that you can use is the optimality-based methods, and these are based around constructing many trees and choosing the optimal one. So you're comparing all of the different topologies, and from some measure of how well the tree matches the distance matrix, you're determining which one of all of the possibilities is the best one. So these have better accuracies than the clustering-based methods because you're looking at everything. But if you have a lot of taxa, and then we're talking in the realms of 8, 10, 12, then they can be too computationally expensive to be feasible to do. There are two of them that we're going to mention, and one is Fitch-Margulies and the minimum evolution approach, and these differ in the metrics by which they determine which tree is the best fitting one. Fitch-Margulies uses a measure that's the square deviation between the distances and the branch length, whilst the minimum evolution is just trying to minimize overall branch length. Then we have the character-based phylogenetic tree construction algorithms, and these consider the ancestral sequences and try to infer them and their likelihood, and thereby determine what trees are the best ones. First off, we have parsimony, and this is the principle of maximum parsimony trees. Parsimony itself, that's the principle of using the fewest resources or explanations to solve a problem, and this is pretty much applying Occam's razor. The simplest explanation is the best explanation. So we're in this method aiming to choose the tree that has the fewest evolutionary changes, so the most parsimonious tree. So to do so and do so effectively, it considers only positions in an alignment which are considered to be informative. So if we're looking at an alignment, we need to have enough changes and the change needs to occur often enough for it to be informative. So in this case, this position could well be informative and something we can continue to look at with this method. So we're constructing all possible trees for this position, so all the different topologies. Then we're going to try to infer what could the internal nodes, so the ancestral sequences, have been, and which option gives us the fewest amount of changes. Starting off with these two trees, we could have either A and A or G and G as our ancestral sequences. These all end up requiring 2 changes to go from the ancestors to our set of observed sequences. If we have G and A as the ancestral sequences, we actually end up worse off, so we require 3 changes. Let's have a look at this tree then. So we have a few different options. Again, with both ancestral sequences being identical, it requires 2 changes. But with this topology, there is one option where we require just a single substitution. So this is the most parsimonious tree for this position. So you look at all of the informative sites like this and see how many substitutions do each possible topology require, and then looking at the full set of informative sites, what could the tree that overall, or the topology and set of ancestral sequences that give the overall fewest amount of substitutions, that's the final tree of this method. One problem that the maximum parsimony method in particular is susceptible to is the long branch attraction systematic error. And this is where taxa appear to be close together just due to the accumulated amount of change rather than due to descent. So long branches end up together with each other in the phylogenetic tree. And here we have an example. So in the true tree, we have a bit of a homoplasy. So taxon 1 and taxon 4 have independently ended up with a G, but the maximum parsimony tree, it doesn't really do homoplasy at all. So it will group these two actually very long branches together with each other just because they're similar, and it will maximize the parsimony. You can alleviate this by allowing for correcting multiple substitution and different evolutionary rate. It can also, to an extent, be alleviated by including more taxa that are related to the longest branches, so you're sort of breaking them up and using more data. You could also choose to use molecular markers that evolve more slowly so that you're not susceptible to this enhanced speed that tend to give rise to this higher rate of change that makes the method susceptible to the long branch attraction situation. We may also want to reduce the number of trees that we inspect to just keep down the computational amount of time needed and make things feasible. One method of doing so is the branch and bound method, and this starts off with building a tree using either neighbor joining or UPGMA. This tree is used to set the upper limit, so like how many substitutions do we need to make this tree happen, or the minimum number of substitutions for this topology. That's the bound. Then we initiate the tree search for the parsimony method, and once a tree variant or tree topology reaches the bound set by the previous tree, then we terminate that path for the topology and carry on only with the ones that are better or equal to. And that's sort of cutting off the bad directions before we spend too much computational time on them. So if you're doing an exhaustive search, you can do about 10 taxa. Using branch and bound, you can do up to around 20 taxa. So an improvement, but still not a huge amount of taxa that you can compare with each other. Another method of reducing the number of trees to inspect is called branch swapping. This starts off with a neighbor joining tree. Then you're cutting off sections of the tree and grafting it to other parts of the tree and checking, was this better or worse than the initial neighbor joining tree? And if it's better, you keep it, and otherwise, you discard it. And then with the tree that you kept, be it the new one or the old one, you do another round of rearranging the tree. And that's how this approach works. And it has a bit of a caveat that it's quite possible to get stuck in local minima rather than actually finding the best tree. So there are always risks involved with these methods, but you do reduce the amount of computation needed to get to a really good tree. Next up, we have the maximum likelihood method. This method aims to identify the tree that has the most likely evolutionary path, and it's looking at everything. It's considering all of the positions in the sequence alignment. It's considering all of the possible tree topologies and all the possible ancestral sequences. So let's have a look at that. So also to get at the likelihood of the tree itself, it's using probabilistic methods that allow you to estimate the probability of a change between things. So one method or one probabilistic model that can be used is the Hughes-Cantor model, and then you have both a probability for a nucleotide remaining the same after a time t and a probability that it has changed. And this depends then on the time that has passed and the nucleotide substitution rate. So the accuracy of the maximum likelihood trees will be only as good as the accuracy of your substitution model. But overall, this is the most rigorous and all-encompassing method for phylogenetic tree construction. Let's have a try at this. So let's look at sequence position 4 and see how we would go through one step of this for one position. So there are 16 possible tree topologies when we have, or 16 possible rooted trees for 4 taxa. This is one of them. So we have at the end our taxa that we know of today, and then there's a number of ancestral sequences that could be either A, T, G, or C. So in the end, we're going to calculate the possibility or the probability for each of these different combinations. The formula for this likelihood for each specific trees is calculated by multiplying the probability of going from whatever nucleotide we have assigned Z to the nucleotide that X has been assigned to. And then the probability of going from X to A, from X to C, and similarly from Z to Y, from Y to T, and from Y to G. So that's what's going on here. Now, multiplication, more computationally intensive than just adding things. And with this situation, if we just go into log and use log probabilities, then we can just add them together. So each of the different possibilities of what X, or the different combinations of what X, Y, and Z could be, they'll have their own likelihood, and presumably one of them will be the best likelihood. Then when you have this for the total tree, you're looking at the conglomerate topology and the different likelihood for your ancestral sequences that align with the topology that you're looking at. So you're summing up your log likelihoods to see what is it all together. Again, this is computationally intensive. We need to reduce the number of trees that we are inspecting. One way of doing this is quartet puzzling. And here you construct many trees with just 4 taxa in them, many subsets, many possibilities. For each of these smaller ones, you construct the maximal likelihood tree, and then using these different quartets, a consensus tree is constructed. Another method is the NJML, so it's a fusion between a neighbor joining method and a maximum likelihood method. So it starts off by constructing a neighbor joining tree. It calculates the confidence for the different branches. You make polytomies if there's low confidence for a branch, and then you resolve what the polytomy should be in a dichotomy using the maximum likelihood method. Next up, there is the approach using genetic algorithms, and this approach is like it has taken inspiration from biology. So it starts off with a population of random trees. Then you're testing the log likelihood score of these trees, and that's sort of their fitness measure. So how good are they? And the highest log likelihood tree is allowed to have offspring with mutations. So branch rearrangements is typically what's used as the mutations to our best trees. And if the offspring is more fit than the parent, it's allowed offspring of its own, and going like that, it continues until it no longer finds a better tree, and quite often you end up with quite a good tree. Finally, there is a Bayesian analysis approach, and that's centered around the Bayes theorem for calculating probability. And this then uses a posterior probability that you're trying to calculate, and that's the probability of the current tree. It uses a prior probability, and that's the probability before you build a tree, and that's you don't know anything about anything, so all of the trees are assumed to be equally possible. There is a conditional probability that you get from the observed substitution frequency of the multiple sequence alignment, and you have the overall probability of the data. In this method, you're sampling the landscape of possible trees using a Markov chain Monte Carlo approach. So it's a hill climbing algorithm that's taking random steps and accepting them if they're good. And it does allow some steps towards something worse, but if it's a very large step towards something worse, that step is discarded. So it's sampling a landscape and progressively getting closer and closer to the tree that has the very best posterior probability. But how good are trees? We need to assess how good are they, our confidence in them. So there are a few methods, there's a number of methods of doing this, and one of the most common one is called bootstrapping. And this is one method for assigning confidence to the topology itself. So the method is that you disturb your data set, your multiple sequence alignment. So we're starting off with an original alignment that will build one tree. But to disturb the alignment, you're getting new variants thereof, and the bootstrap way of doing this pertursion is to make a new alignment that's the same length as the original one by randomly taking different columns from the original alignment. Having done this, we can build trees from our perturbed alignments, from which we can make a consensus tree where we can add our bootstrap values, so the percentance of appearance of that piece of topology in the bootstrap replicates. This is something that we need to do lots and lots of times, so we need to have many separate replicates of perturbing the alignment and building trees. So it's recommended that it's around 500 to 1,000 replicates to be sure that you have good bootstrap values. And this allows you to see how robust a section of the tree is. So how well does it withstand fluctuations in the input data. So it's telling you something about how stable is a part of your phylogenetic tree. Another method that's, well, it's a different way of perturbing the dataset, but it's in essence telling you the very same thing as bootstrapping, just a slightly different way of getting there. It's jackknifing. So here we take our original sequence. We're randomly deleting half of the columns, so we end up with something that's half as long, but randomly kept half of them. We construct trees from our replicates. Again, you probably want to be having loads of them. From that, we can get a consensus tree with a jackknife value that's essentially the same sort of value as what we had for the bootstrapping method. The final thing I'm going to mention here is the Bayesian simulation approach for assigning confidences. This is something that falls out from the Bayesian analysis tree construction method, and you're essentially using your sampled trees during the algorithm to estimate your confidence in the different parts of the tree. And these trees tend to be better because you're progressively going there, so your sample trees are closer to the actual trees than what you're getting from disturbing the alignment in bootstrapping or jackknifing. So the actual bootstrap values then tends to be higher than your bootstrapping values, and they will have different statistical meaning. So don't compare bootstrap values with values from Bayesian simulation. Finally then, how do we choose what method to do when? Now, it will depend on your data set, what you're looking at, what you're trying to do, your computational methods available, but there are a few useful rule of thumbs for when something is suitable. So if you have a strong sequence similarity, then maximum parsimony methods can be a good thing to choose, because you're probably dealing with something that's not too evolutionarily distant from each other, and then the assumptions of maximum parsimony can still hold true. If you have clearly recognizable sequence similarity, so like it's not super close, but you can still definitely tell that these are closely related to each other, then distance methods are quite often the appropriate choice, especially if you're dealing with lots of sequences. Whilst the maximum likelihood, that strength can give you the computational investment may then be not as appropriate if you have clearly recognizable sequence similarity, but you do need that extra resolution and rigor if the sequences are very different and hard to get that with any of the other methods. That's sort of the material that we're going to go through today, but let's have a little look at what's going on in our wide world today. So if you haven't noticed, there's been a little bit of a pandemic with the COVID-19 disease caused by the SARS-CoV-2 virus. Now, one thing that was clear quite early on was that it's quite likely that the SARS-CoV-2 virus originated in bats, so it spilled over from bats probably to an intermediate host, and then from that intermediate host spilled over to us humans. How do we know that it's from bats? Well, phylogeny and phylogenetic trees to compare the evolutionary history and look at how similar are things to each other. And we can quite clearly see that SARS-CoV, the original SARS, cluster together with bat coronaviruses, as do the SARS-CoV-2 virus. So this is from a paper that's old enough that we had the 2019 novel coronavirus name for it. That clusters most closely to bats in this study, and it's surrounded by quite a lot of bats, but still, of the human coronaviruses, it ends up being closest to the SARS-CoV-1 virus. And with that, I hope that you have enjoyed learning lots and lots about phylogeny, and thank you very much!